Hello, A&P1 students. Gina Piscatelli here with our last integumentary system lecture. This will just focus on homeostatic imbalance, and specifically burns and skin cancer is what we'll talk about. So the causes of burns, I'm going to start with burns, include heat, which really usually is a flame, but it could be just intense heat, electricity, and radiation, including UV radiation, as well as some chemicals can cause burns to the skin. Now, what's the problem with the burns? Well, obviously it kills the cells, right? So then you have to repair the tissue, and it hurts. But there's also some more serious, well, if it somewhat more serious consequences, and that is if the burn is severe enough, a person can lose body fluids, proteins, and electrolytes, which could have secondary problems. In addition, the dead tissue is a feeding ground for microbes, so infection is a risk. And then finally, as the tissue repair process begins, fluid is sent to the site of damage, and therefore you have a reduced blood volume. That can lead to circulatory shock if it's severe enough. So as you can imagine, burns damage the layers of our skin, starting with the epidermis, which is usually the point of contact, and might penetrate the dermis or even go below the skin into the hypodermal tissue or subcutaneous tissue. We classify burns based on how many layers or how deep the skin is affected, how deeply the skin is affected. A first-degree burn involves only the epidermis, so only stratified squamous epithelial cells are damaged. You'll have localized redness, swelling, and pain. It tends to heal in 2 to 3 days, I guess. Sunburn is usually a first-degree burn, but I've seen some sunburns that were almost second degree, believe it or not. Second-degree burns involve all of the epidermis and the superficial part of the dermis. So that's what's injured, and redness, swelling, pain occurs as in first degree, but also blisters may form. Usually there's little or no scarring as the tissue regenerates, but it takes a while, 3 to 4 weeks. During this period, it's important to keep the area clean and prevent infection. And that's because the dermal blood vessels have been exposed and damaged, and infection could travel throughout the body. And then a third-degree burn involves the epidermis and the entire dermis, so all the way down to hypodermal tissue. The burned area usually appears gray or white, but it can also be red or black. In the picture shown here, it looks black. Nerve endings are destroyed, so initially it's not that painful, but as regeneration takes place, it becomes quite painful. And regeneration takes so long that usually skin grafting is performed. Another way that we can classify or describe burns is to describe the extent of the surface area of a patient. How much is affected. And to do this, we usually use the rule of 9s. And the body surface is divided into areas representing 9% or multiples of 9. So notice the entire head, 9%. The anterior thorax, 9%. The posterior thorax, 9%, etc., so on and so forth. Palms are usually 1%, and then you add those up. So you might have heard something like a case where a patient had 3 third degree burns over 45% of his body. I mean, that's horrible, life-threatening, right? So there's different ways to describe it. The most life-threatening problem or effect of a burn would be fluid loss. That initially. So first you've lost the barrier, and water evaporates, and you've damaged blood vessels. Now we're talking about severe burns here, but you've damaged blood vessels, so you'll probably have blood loss. But that will, blood volume will go down even further as the repair process takes place. So fluid will be transported to that site of injury for repair, and the decrease in blood volume can cause the heart to go into arrest, so cardiac arrest, or you can have what is known as circulatory shock. There's just not enough blood to pump. So that's first, you have to replace the fluid, right? The second most life-threatening effect of a burn is infection, and that's, as I've already mentioned, the fact that the dead tissue quickly becomes infected by bacteria and fungi, and if either of those enter the circulatory system, then you can get a whole body-wide infection. And a syndrome called sepsis could occur. Sepsis is a widespread inflammatory response to infection, and it can lead to organs malfunctioning, but ultimately to their failure, so they can no longer function at all. So treatment is centered around three things. One, supplying the patient with enough nutrients to replace the proteins that they've lost, the fluid that they've lost, and support tissue repair. Nutrients. I've heard it said that a burn patient can't eat too much. It just takes that much energy and nutrients for repair. And then secondly, of course, we have to hydrate. Usually that's done with an IV fluid. And you have to make sure that hydration includes the electrolytes that are required for osmotic balance of the blood and the tissues. And then the third thing is that infection needs to be prevented. So antibiotics are a routine treatment. And to prepare the tissue for a skin graft, or to get rid of the dead tissue that's a feeding ground for bacteria, they can do a procedure called debridement. I don't know if I'd call it a procedure, but they have to remove the dead tissue. So kind of peel away all the dead tissue. Evidently, by the time they get to that stage, it is quite painful to patients. But then a skin graft is usually what needs to happen in a severe burn treatment. Okay, the next topic besides just burns is going to lead into skin cancer. Okay, but one of the major causes of skin cancer is exposure to UV radiation. So UV radiation causes burns, sunburns, right? And I want to tell you two things that are good about UV radiation and two things that are bad. So first, the good things. UV light stimulates our skin cells, keratinocytes, to make vitamin D. And we need vitamin D for normal bone maintenance and growth, but maintenance. So that sounds like a good thing. We need UV light to make vitamin D. The second good thing is that UV radiation stimulates the melanocytes in the skin to produce melanin. Well, that melanin shields our DNA, so the DNA in the cells won't be damaged. And of course, it produces a suntan, right? Because it's a pigment. Eventually, the suntan fades because the melanin is degraded. Now, there's two things that UV radiation does that are terrible. One is it breaks down folic acid, which is vitamin B, and that is needed for normal cell division and for fetal development. And secondly, UV radiation can lead to skin cancer because it alters DNA. And if the DNA is altered in a cell, the cell probably won't be functioning normally. Sometimes the DNA is altered in such a way that cell division is instigated, I guess, or stimulated, and it shouldn't happen. So you get proliferation of tissue, and it consists of all these mutated cells that aren't functional. So two good things and two bad things. Cancer is actually unregulated cell division of mutated cells. So you can have a group of cells undergo mitosis, even though they don't really need to, and form a mass that we might call a tumor, but it could be benign. It might get in the way, but it stops eventually, and the tissue doesn't hurt anything, so that's benign. But in cancer, you get unregulated cell division of cells that are abnormal. So it's going to continue to grow and grow and grow and grow, and the cells are not going to function correctly. And this is all at the expense of normal tissue. So, with that in mind, what cancer is, let's look at the three types of cancers associated with the epidermis. There's basal cell carcinoma, squamous cell carcinoma, and melanoma. Melanoma is the one that I hear the most about because it's the most deadly and aggressive. But it's really the least common. The one that's the most common and easily treated, I don't know about easily, but has an excellent success rate, is basal cell carcinoma. So that's the most common, and it's the least troublesome because it doesn't metastasize, which means cells don't break off and move to another part of the body. Whereas melanoma definitely metastasizes quickly. Now, melanoma, even though it's the least common and most deadly, I guess most deadly is what I meant to say, if it is detected very early in one localized spot, there's an excellent success rate after it's removed. And I have stats there, 99% of people survive at the 5-year point. I don't know beyond that. Okay, so squamous cell carcinoma is not as common as basal cell carcinoma and not as rare as melanoma. If you have early detection of that, there's also an excellent survival rate. So let's see if we can tell the difference and talk about what kinds of cells are involved. So basal cell carcinoma is happening in the stratum basale, where there are stem cells that are already dividing and keratinocytes that are working on dividing or have just divided and they're maturing. So basal cell carcinoma is the most common because that's where cell division is already taking place. Squamous cell carcinoma is of the cells that are dividing are in the stratum spinosum usually. And melanoma, it's the melanocytes in the stratum basale that are undergoing unregulated cell division. And there are pictures of the different types here, so they correlate. They go in the same order. So the top one is basal cell carcinoma, the middle one is squamous cell carcinoma, and the bottom one is melanoma. That's the only one that I can make sense of because, it looks so dark. Obviously, there would be a lot of melanocytes, which are pigmented, right? So just to protect yourself, always get anything suspicious looked at and follow this rule, the ABCD rule. Look for asymmetry, meaning it's not a complete circle like a typical mole or freckle. Look at the border and see if it's scalloped. And then look at the color, which makes me laugh a little bit because all of those were different colors, each type of skin cancer. And if you read most texts, they'll tell you it could be brown, black, tan, red, yellow. That's a lot of different colors, but still, if it changes, check that out. And then the diameter. If it's greater than 6 millimeters, especially if it started small and it got up to 6 millimeters in diameter, you should have it checked out, and they'll probably excise it and send it off to pathology for somebody to look at. Thank you. That's the end of Unit 2, as well as the end of the integumentary system.